Michaela, you may not have logged on when I said this, but it's not linked up yet, but by this afternoon, I should have a link to a playlist on YouTube where these presentations are going to be recorded. So anything you might have missed, hopefully you'll be able to download. The very first is not going to be up there, but the rest will. Okay, like I was saying, I remember to press record. Okay, I did. Okay, as I was saying, make sure you get your knowledge of Mendelian genetics back. Because I'm going to assume from here on out that you remember that genes exist in one or more variant forms called alleles. For any given gene, one allele may be dominant, which doesn't mean it's superior or better, but it does mean that if you have a dominant and a recessive, that means you're heterozygous, and the dominant gene is the one that's expressed in the phenotype. So I'm going to assume that you remember dominant and recessive alleles, that you remember that sometimes alleles of a gene might be independent or incompletely dominant, like the gene for white spotting in kitty cats, or might be co-dominant, like human blood groups, and so on. Make sure you remind yourself of how all that works, because we're going to be basing it, what's to come, on that. So we can start using math and applying it to the study of evolution. If we start calculating numbers called allele frequencies. And all this is the sum of all copies of an allele in a population divided by the sum of all alleles. Okay, let's take a human genetics is weird because so many traits are actually polygenic, but one of the few human genes that is, or human traits that's controlled by a single gene with two alleles, so it fits Mendelian rules very neatly, is the consistency of your earwax. Some people have moist earwax. Some people have crumbly earwax. And I think moist is dominant to crumbly. And here we are in this room, we are 5 people. Yes. Each one of us has 2 copies of an allele for earwax consistency. Right? Yes. You got one from your mom and one from your dad, and you'll pass on one of your earwax genes to each of your offspring if you have it. Right? So if there's 5 people and each one of us has 2 copies of the gene for earwax consistency, then we have 10 alleles in all, right? 5 people times 2 alleles per person. So in principle, we could try to figure out what everybody's alleles were. And let's say we have 2 people that are homozygous for wet earwax and 2 people that are homozygous for crumbly earwax and 1 person that's a heterozygote. Well, that means we have 5 alleles for moist earwax and 5 alleles for crumbly earwax. Okay. When a gene has just 2 alleles, we can usually indicate those frequencies. We can count, any allele frequency is a number between 0 and 1. All right. We can also represent it as a percentage if you want to, and I assume you can easily go back and forth between percentages and decimals. And when a gene has 2 alleles, each allele has a frequency, and we abbreviate those frequencies as P and Q. And P plus Q always equals 1, right? for the same, because, these are decimals that can also be percentages. If 70% of the alleles in this room were moist earwax and 30% were crumbly earwax, right? 70% and 30% must always add up to 100%, right? All your percentages have to add up to 100. Okay, here we are. All right, good morning, Aaron, joining our merry band. Okay, so modern synthesis evolutionary biology starts by calculating allele frequencies in a population and looking at whether they change. Let's say that for Aaron, who just came in, I've been using a human gene that is known to have only 2 alleles. The consistency of your earwax is governed by a single gene with 1 allele for moist earwax and 1 allele for crumbly earwax. And if, let's say, having moist earwax gave you some kind of advantage, maybe it made you less prone to ear infections or something like that. I don't know that it does, but let's just pretend. If having moist earwax meant that you were less likely to get ear infections, and that means, better able to keep your sense of hearing and, less prone to get ear infections that can spread into your skull bones, which they sometimes can, then maybe more people, or maybe people with moist earwax would have greater probability of having kids than people with dry earwax. And we might expect to see the frequency of the moist earwax allele increase over time. On the other hand, if dry earwax gave you some sort of increased odds of not just surviving, but reproducing, then maybe the frequency of the dry earwax allele would go up over time. And we can look for things like this. A quick note before I get into a little more math is that allele frequencies are sometimes used as indicators of a population's past history. Sometimes events that happen to a population leave traces in allele frequencies themselves. This is basically the kind of thing that those groups like 23andMe do. Right, where you send off a DNA sample and it turns out that you're 0.012% Viking. Have you seen those commercials? The guy keeps waiting and waiting for his DNA results, finds out that he's 0.012% Viking, and promptly goes out and buys a Viking sword and a horn helmet and starts setting the neighborhood on fire, right? Which, by the way, is really dumb because Viking was not an ethnicity, it was a profession. That would be finding out that your DNA is 0.012% plumber or 0.012% office manager or something like that. Vikings were not an ethnic group at all. But anyway, what they can sometimes do is find human genes that, and there's not that many of them, but there are a few human genes that happen to be very common in limited numbers of people, in limited ethnic groups. And if you have one of these genes that's strongly correlated with an ethnic group, there's a high probability that you're descended from that ethnic group. It's not really as accurate as people think it is, but you can at least get a rough idea of where your ancestors might have come from if you do this. And again, they can't pinpoint you down quite as accurately as they might like. And like my dad went from 90% to 70% Northern European just because they updated their database and changed some of the data they were using to make these estimates. So it's not really that cut and dry, but you can at least get a something of a look into the history of people and populations by using certain allele frequencies. A little bit more accurate is that we can look at the frequencies of alleles that only appear in one copy per person. The vast majority of genes in your genome, you've got two copies of, right? You've got one from your mom and one from your dad. And you'll pass on one of each gene to each of your offspring if you have any. But there's two major pieces of DNA in the human body that only appear in one copy per person. Almost all the time. There's rare exceptions. One of them is the DNA present in your mitochondria, right? Everybody's got mitochondria, and you always inherit your mitochondria only from your mother. So you got your mitochondria from your mom. Those of you hearing my voice who are female, you will pass on your mitochondrial DNA to all of your offspring. Those of us who are male, we won't. I did not pass on any mitochondrial DNA to my son. So mitochondrial DNA is one piece of DNA that only appears in one copy, and it is passed on through what's called uniparental inheritance. It only gets passed on through one parent. Mitochondrial DNA is only inherited through the mother. The other one is Y chromosome DNA. If you're a man, you got your Y chromosome from your dad. You did not inherit your Y chromosome from your mom, because if you had, your mom would be your dad, right? It's the Y chromosome that contains that genetic switch that starts you developing male plumbing. Question?
Yes, is there a specific reason of why the mitochondrial genes come from your mom?
Because a sperm cell is basically very tightly packaged DNA and a flagellum, and that's it. There are some mitochondria in the shaft of the sperm cell, but there's none in the cell body. And normally the part that's got the mitochondria does not penetrate into the egg. There may be a few rare cases where it does happen, but you'll occasionally get that, but it's very rare. And then of course, Y chromosome DNA, a father will pass on Y chromosomes to all of his sons. Mom, of course, does not pass on Y chromosomes at all, because if she did, mom would be dead. You can occasionally find people who are anatomically female but have XY chromosomes. They're usually sterile, and that gets a little bit far afield. There are people with intersex conditions that can look and feel female but have Y chromosomes, but they usually don't pass them on because human biology is really cool that way. Anyway. So there are some alleles that are only passed on uniparentally, and the only thing that causes uniparentally inherited DNA to change is mutation. DNA that's inherited biparentally can be changed by recombination as well, right? Remember recombination? It's all that crossing over that goes on at the beginning of meiosis. I got one copy of chromosome 1 from my mother and one from my father. What my son inherited from me is a copy of chromosome 1 in which my mom's chromosome 1 and my dad's chromosome 1 have been spliced together several times, right? That's how recombination works. So my son's chromosome 1 is not identical to either one of mine. Because DNA gets shuffled during that process. But that doesn't happen with DNA that's inherited uniparentally. And when you look at this DNA, you can find variation in that uniparental DNA, and you can classify that variation into what we call haplogroups and haplotypes. Remember, haplo means having only one of something, as in being haploid, right? Well, just as your genotype is what we use for what alleles you're carrying in most of your genes. Your haplotype is what alleles you're carrying in your mitochondria and on your Y chromosome if you've got a Y. Those are your haplotypes. So they're those unique sequences that are found on genetic entities that rarely or never recombine. The big ones are mitochondria and Ys. And the only thing that causes a haplotype to change is a mutation. So barring mutation, I've got the same Y chromosome as my dad. And he got it from his dad, who got it from his dad. And so I've inherited my Y chromosome from my father's father. And the only thing that has changed that Y chromosome has been the occasional mutation. And I got my mitochondria from my mother's mother. And the only process that has caused that DNA to change has been the occasional mutation. And if it should happen that you and I happen to share the same Y chromosome or a very similar one, that must mean that we have the same father.
Yeah.
Yeah, or, for all I know, we could be 10th cousins or something like that. It doesn't have to go back all the way to Neanderthal times. But yeah, we could establish that we are related through common ancestry if we were to compare our Y chromosomes. And the most common types of haplotypes, there's SNPs, which stands for single nucleotide polymorphisms, which just means that at position 23,795, my Y chromosome might have an A and yours might have a G. Okay, that's just a single nucleotide difference in Y chromosome DNA or mitochondrial DNA. Maybe at position 8,176, my mitochondria has a T, your mitochondria has an A, or something like that. You'll also see these called UEPs, unique event polymorphisms. And the ones that we use for tracing evolutionary history are not supposed to be functionally important. They're not supposed to make any difference in survival. They're just there. So if we've selected them properly, then those differences are not the kind of thing that will be affected by natural selection. It's completely neutral variation. The other type of haplotype is STR, aka single tandem repeats. There are areas in your DNA, usually not part of functional proteins, although sometimes they are, where you'll just get part of your DNA is made up of a 2 to 6 nucleotide sequence that's just repeated over and over. So there might be some spot on one of my chromosomes where one of the strands is CCG, 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 and then eventually it'll go on and do something else. Those are short tandem or single tandem repeats. So for all I know, at some position on my Y chromosome, I might have 10 repeats of AAGC, and you might have 12 repeats of AAGC. Or my mitochondria might have 5 repeats of GGT, and yours might have 9. And in essence, yours might have 12, and maybe yours has 3. And again, this variation is not supposed to be selectively important. It generally makes absolutely no difference whether you've got 12 or 9 or 3 repeats in one of these single tandem repeat sequences. There are cases where these can pop up in the middle of important genes, but the ones that we use for tracing are not supposed to be functionally significant at all. They're just there. And if my mitochondria have 5 GPT repeats and yours have 5, it is possible that we share a common mother, which could go back all the way to the time of the cavemen, or it could mean that we're actually 8 cousins 3 times removed and just didn't know it until now. So, you know, I don't know if you got any ancestry from North Carolina, then it's possible. Who knows? Okay, right. So that's a map of some Y chromosome single tandem repeats. There's a diagram of the chromosome itself on the bottom. The single tandem repeats are given these code numbers. And again, they're functionless. They don't affect what the chromosome does. Not that the Y chromosome does very much other than trigger the developmental process by which you grow a tallywhacker. But these don't affect that chromosome function. They're just minor differences that just happen to be there. Okay. Oh, question coming up on the chats. Okay, Michaela. All right, we're recording this, so if you have to step away from the chat, you can watch it at some other time. Good luck and hang in there. Go do what you need to do. All right, I'm checking the chat one more time. Okay, do your thing. This happens to be a world map of population mitochondrial haplotypes. And there's lots of different haplotypes, but they're grouped together into similar haplogroups, which have been given code letters. So, for example, like up here in North America, these are Native American populations, by the way. And these 2-letter abbreviations are abbreviations for names of tribes or ethnic groups or what have you. And you can see most Native American and Native South American populations have mitochondria with haplogroups. Looks like it's mostly, okay, haplogroup A is that vibrant purple. Haplogroup B is that strong red. Haplogroup D is that solid blue. So it's mostly A, B, and D, and it's kind of covered up here, but that's some of haplogroup C as well. And again, it doesn't really tell you anything about Native Americans other than the fact that Native Americans come from a relatively limited number of mothers, mothers, that have ultimately passed on mitochondria to their descendants. If you go over here and look at Europe, this is the ancestry of several of us, although clearly not everybody. Most European mitochondrial DNA, there's actually a lot of haplogroups represented, but easily about half of it in the typical population is haplogroup H. And again, these are not what make races different from each other. Native Americans don't look different because they've got these haplogroups. They're just there. They're not thought to have any function. The reason this might be interesting is that, let's say you wanted to know where Native Americans came from. Well, the leading hypothesis for a long time has been that they're descended from Asian populations that migrated across the Bering Strait from Siberia to what's now Alaska. And they used to say this happened 11,000 years ago. There's new archaeological evidence that Native people have been here longer than that. I don't think we really yet know exactly how long, because some of the evidence has been hard to date, but it's at least 14,000 years, probably more. But what we can do is look at Siberian populations and see if any of them have similar mixtures of mitochondrial haplotypes. And so, for example, like look for the zones that are purple, that match the purple right there. That's actually fairly widespread, but we've got a fair number of purples over here in a population called the Kets, and a population called the Komi, and a population called the Altaians. The Altaians, we've also got a fairly high number of that blue right there. That's haplogroup D, and that's also one we find among Native Americans. So it's not conclusive, but we can at least try to start identifying populations that might be distantly related to present-day Native Americans. Okay, you with me there? Yes.
And is that red involving the same red that's in the Americas?
Yeah, evidently it is. That red also seems to be found in more in South Asia populations. So in the Mongols, for instance, that MO right there seem to have it. So maybe there's not one single origin for all Native Americans. And I mean, certainly the Altaians and the Mongols today have changed from what they were 20,000 years ago. And there's a lot more sophisticated types of analyses that you can do that I'm just not going to go into. It actually starts getting out of my depth. But that's what mitochondrial diversity looks like. And that happens to be a world map of Y chromosome haplotype diversity. So most natives have haplotype Q. Western Europeans have a bit more diversity, but a lot of them have haplotype R1b. That's what my dad has, and I've never had my own tested, but I assume it's what I've got. And you can just see, a lot of South Asians have haplotype O, and yeah, there it is. What's up?
Just I found this really fascinating. You you'll post slides.
Yeah, I will.
Okay, sorry. Yeah.
Yeah. Oh, one thing you might note is there's an ethnic group up in Siberia with a very high frequency of, no, that's A, not Q. Darn it, the colors are so close. But I think most Native Americans have Y haplotype A, and there's a high frequency of it up here in this group called the Selkups. And the reason that's cool is that the Selkup language is the only one so far that is plausibly anything like certain Native American languages. We can actually make an argument for relationship because Selkup and a related language nearby called Kit is actually somewhat similar to Navajo. With most of those languages, there's just been such a long time. Native American languages have changed so much that we cannot reliably, we can't link them to any languages spoken outside of North America. But we can make a case for Selkup being similar to Dine Bizan, Navajo, and related languages there. So that's one case where we can actually back some of our DNA data up with some cultural data. Here's a slightly smaller example. There's a set of closely related Y chromosome haplotypes called C3 star. All that C3 star is just a collection of single nucleotide polymorphisms and short tandem repeats. And it turns out that not just C, but C3 star is shared by 8% of all Asian men, which works out to about 0.5% of the world population. And in some North Asian populations, notably the Mongols, If you look at Inner Mongolia, up to 25% of all men have the C3 star Y chromosome haplotype. Now, if two men share Y chromosome haplotypes, it means that they have a common father, father. And in this case, looking at the mutations that went into creating C3 star, we can actually get a rough estimate of when that common male ancestor lived. And we even have we've even been able to make a case as to who that common ancestor was. You heard of this? You're nodding. The likeliest father, who passed on his Y chromosome to 1/20th of the population of the world seems to be Genghis Khan. Now, Genghis Khan was the emperor of the Mongols. The Mongols came storming out of Mongolia and ended up conquering an empire that went all the way to Russia and all the way to the Middle East and encompassed most of China, that basically spanned very large amounts of most of Asia and a sizable chunk of Europe at its height. Have you heard of these guys? Okay, enormous armies mounted on horses, some of the greatest horsemen the world's ever known. And Genghis Khan was the emperor of the Mongols who created this enormous empire, one of the largest in world history. And supposedly Genghis Khan said the greatest joy for a man is to defeat his enemies, to drive them before him, to take from them all that they possess, to see those they love in tears, to ride their horses, and to hold their wives and daughters in his arms. And yeah, I'm not really comfortable talking about this, but when he says hold their wives and daughters in his arms, he doesn't mean giving them sympathetic hugs, okay? Genghis Khan, raped his way across a whole freaking continent and must have had a ridiculously large number of offspring. So Genghis Khan very enthusiastically spread his Y chromosome as often as he got the chance. And aside from that, he had 4 sons by his primary wife. A lot of the people he had kids with, he wasn't exactly married to. And he had a whole bunch of wives. He had 4 sons, this is them, by his primary wife. And after Genghis Khan died, to claim power in the Mongols, you had to be descended from one of his 4 sons. You had to be part of what was called Altan Urag, the golden lineage. And of course, each of these 4 sons had many wives and probably also did their share of rape. And their sons would have taken many wives and et cetera, et cetera. And so when you have this very successful man who spreads his Y chromosome everywhere you can get it, and his sons do the same, and his grandsons do the same, and that Altan Urag lasted until, I want to say, the 1930s, was when the last official descendant of Genghis Khan finally died. It may not have been the 30s, but it was in the 20th century. Multiplied with all of the illegitimate offspring that got fathered, then it's perhaps not surprising that 1 in 20 human beings, 1 in 200 human beings on the planet are carrying Genghis Khan's Y chromosome DNA. So that's the kind of insights into history you can get when you look at haplotypes and you look at haplotype frequencies. There are some other cases. There's an awful lot of Irishmen who have a particular Y haplotype in common, and it's been suggested they're all descendants of a legendary Irish king named Brian Boru, who had lots of wives and probably some knee tremblers, and would have fathered lots of offspring who would themselves have gone on to have lots of offspring. There's several cases where you can find a very common Y haplotype plausibly linked to a very energetic male line ancestor. So yeah, it happens. Okay, and thereby that's the spread of the Mongol Empire. Genghis Khan's C3 star Y chromosome is thought to have spread over all of North Asia. You can see where they went. They grabbed North China. They would actually move into South China later, all over Central Asia. You can see that they went as far as Kiev. Russia was actually ruled by the Mongols, but the Mongols had to get some of the locals to run the local government. And the minor officials that ran this dinky little town in Russia called Moscow ended up being very successful at gathering power for themselves. But it wasn't until I think it was Ivan IV who finally got rid of every trace of Mongol rule. Ivan IV, by the way, was Ivan the Terrible. Although terrible doesn't mean he was bad, it means he was scary. Ivan the Fearsome. Right. I digress. Okay. There's a problem with using most genes because each individual, for most genes, everybody has 2 copies. And of course, dominance mask recessives. Mendel worked out that flower color in peas was governed by a single gene with 2 alleles, purple and white, right? And we can write those as big P and little p, yes? So a white flowered pea has 2 copies of the recessive, and we can write that as little p, little p. A purple flowered pea, we can't tell whether it's homozygous dominant or it's heterozygous, right? It could have 2 copies of the purple allele and have the genotype big P, or it could be heterozygous and have the genotype big P, little p, and we can't tell by looking. So if we have a whole bunch of purple flowered pea plants, it is possible that all of these purple flowered pea plants have 2 copies of the purple allele, in which case the frequency of the purple allele would be 1. It's possible that all of these purple flowered pea plants are heterozygotes, in which case the allele frequency would be 0.5. Or it could be anything in between if it's got a mixture of heterozygotes and homozygotes. So we can't calculate the allele frequencies simply by looking. Enter G.H. Hardy. Hardy was based at Cambridge University. He was a mathematician. Most of his work was done in fields of mathematics that you have to be in grad school to work with. Most of his work was in a very abstract field called number theory. And again, this is not something you're likely to have been exposed to. Maybe you get some number theory as a senior math major, but it's pretty abstract and pretty out there. And Hardy did most of his work in that field. But he had a friend, a biologist at Cambridge University, whose name was Reginald Punnett. And if you've ever done Punnett squares, that's why they're called Punnett squares. They were worked out by Reg Punnett. And Hardy's other passion, other than mathematics, was cricket. Cricket is this British game that is a lot like baseball, but it takes a hell of a lot longer. And the bat is flat, and there's the added wrinkle that the pitcher is trying to knock these pieces of wood off of stakes with his pitches that the batter is trying to hit. And there's a playing position called silly mid-off. Cricket is just weird. Any of you guys ever seen it or heard of cricket?
There's a documentary on Netflix
Mhm.
It's about everything. Explain like 20 minutes of a lot of different stuff and one cricket.
Okay, all right.
It's very complicated.
I assume it's not that popular in Brazil. It's the only place in Conway I've seen people playing cricket has been on the TV of that Indian restaurant back before the pandemic. I don't know if they still do. There used to be some folks that would come out and play at the park where I take my kid because one of them had, I think, one of them had spent some time in India and had developed a taste for it. But yeah, it's not super popular over here. Anyway, so Hardy and Punnett were crazy about cricket. And one day, probably, at the end of a exciting cricket match that had taken 12 hours, Punnett happened to mention to Hardy that we have this problem of how do we calculate allele frequencies. A guy by the name of G. Udney Ewell had claimed that in any population, dominant alleles would always become more common. Hardy immediately saw this wouldn't happen. And in 1908, he knocked off a short letter to the scientific journal Nature. And the math here is not number theory. The math is basically high school algebra, and he probably just knocked off this letter over breakfast or something. He probably wrote it between his soft-boiled egg and his toast, explaining what exactly would happen. And he documented something that we now call Hardy-Weinberg equilibrium, because a German guy named Wilhelm Weinberg had a similar idea at about the same time. To be fair, there was an American who came up with the idea at about the same time. So if we want to cheer for America because USA, USA, you could call it Hardy-Weinberg-Castle equilibrium, because the guy's name was Dr. Castle. But most people just call it Hardy-Weinberg. So if you have a population that has two alleles of one gene, we can represent the frequencies of those alleles as P and Q. Now, because the frequencies of alleles always add up to 1, so P plus Q is always going to equal 1. And therefore, if you only know P, you can always calculate Q, because P is always going to be equal to 1 minus Q. Q is always going to be equal to 1 minus P. I've given test questions where I've asked, what is P plus Q, and have students pull out their calculators. You don't need to do that. P plus Q is always going to be equal 1. Hardy worked out that if your population meets certain criteria, then the genotype frequencies will be as follows. The frequency of the homozygote for the first allele, call it big A, is going to be P squared. In fact, more generally, the frequency of any homozygote in the population is going to be the square of the corresponding allele frequency. So the frequency of big A individuals is going to be P squared. The frequency of little a individuals, homozygous for the other allele, is going to be Q squared. And the frequency of heterozygotes is going to be 2PQ. Okay, you with me there so far? You might notice that if you add up P squared plus 2PQ plus Q squared, you get 1, because P squared plus 2PQ plus Q squared is what you get if you square P plus Q, right? And P plus Q is always equal to 1. So if you square P plus Q, that's equal to 1 squared, which is 1. But nobody took their calculator out to answer that. So if you know the frequencies of your alleles, you can calculate the expected frequencies of genotypes. If you know the frequencies of genotypes, you can calculate the frequencies of alleles. That means you can do things, and we're going to start doing them on Monday. You can do things like, if I give you data on allele frequencies, you can calculate what the genotype frequency should be using the Hardy-Weinberg relation. And then you can go out and see if the real genotype frequencies are the same as what you calculated, or if they're different. By the same token, you can, if I give you just the frequencies of the genotypes, you can estimate the allele frequencies from that. And starting Monday, this afternoon, I'm going to put up some practice problems for you. You don't have to do them today, but they are the kinds of problems that I give on exams. And they will, they're examples for how you can apply Hardy-Weinberg theory. If a population fits certain criteria, then first of all, the allele frequencies and the genotype frequencies are always going to be related. And if you know one, you can calculate the other. And Hardy also showed that if the population fits certain criteria, then the genotype frequencies will not change from one generation to the next. And the allele frequencies will also not change from one generation to the next. This state, by the way, where genotype frequencies don't change and allele frequencies don't change, is called Hardy-Weinberg equilibrium. Now, here's the thing. A population in Hardy-Weinberg equilibrium doesn't have any change in allele frequencies at all. If we go out in nature and we see that allele frequencies are changing, that's interesting, because one of the conditions for Hardy-Weinberg equilibrium must be being violated. That means something interesting is happening, and we can try to figure out what it is. So the criteria are, first of all, the population has to be large. For perfect Hardy-Weinberg, the population would have to be infinite. And of course, we never get that. But the population must be big. And later next week, I'll be able to talk about just how big is for a population to be in or close to Hardy-Weinberg equilibrium. We'll get more rigorous about that later. There can't be any selection going on. So no natural selection, no artificial selection. There can't be any mutation going on. There can't be any migration in or out, and mating has to be random. Incidentally, by random mating, I don't mean mating without anybody knowing who it even is, okay? I don't mean what everybody claimed went on in the Pi Kappa Alpha house when I was an undergrad, although I'm sure 99% of it was just hype. I don't mean anything like that. Although in some species you would get that, like in oysters, males and females just fire off big clouds of eggs and sperm right into the water. And, you know, which egg meets which sperm is pretty much a random draw. But random mating basically means you have no sexual selection, you have no inbreeding. Individuals are not choosing mates in any way that would bias the allele frequency. Here we go. Let's take Valonia. All right, town of about 5,000 close to here. And let's say that everybody in Valonia is a good Baptist and follows strict rules of morality, and everybody in Valonia marries their childhood sweetheart and is completely faithful, and there's never any adultery, and all marriages last a lifetime, and everything is just perfect in Valonia. Okay. The real Valonia is probably not quite like that, but pretend for a minute that we're in Baptist utopia here. You could still have random mating. How? There's actually multiple genes that govern eye color. So don't take this too literally, but very roughly, brown eyes are dominant to light eyes. except for the cases when they're not because other genes get in the way, but work with me here. If having brown eyes or blue eyes makes no difference in whether or not you get to marry and have kids, then we would have random mating with respect to eye color. Like if all of the blue-eyed girls in Valonia are having to fight off gentlemen callers with a stick, and all of the brown-eyed girls in Valonia are sitting at home on Friday nights doing nothing, because like, what would they be doing in Valonia? Then that would be non-random mating. But if having blue eyes or brown eyes makes absolutely no difference to whether or not you get married and have kids, you know, if it's completely irrelevant for mate choice, then we could say that in the population of Valonia, we have random mating with respect to eye color. Okay, are we happy there? And it does not mean that the residents of Valonia are just picking their wives by drawing out of a hat or anything like that. And it doesn't mean that everybody in Valonia is a total swinger. It just means that you don't have sexual selection. There's some other things that can violate the random mating criterion, like inbreeding, for example, will do it. Insert joke here. But there's a lot of plants that will self-fertilize oftentimes, and self-fertilization is going to throw this off. So if a population meets all 5 of these criteria, then the genotype frequencies will be linked to the allele frequencies, and neither the genotype frequencies nor the allele frequencies will change from one generation to the next. The reason that's interesting is what we can do is go out and measure actual allele and genotype frequencies and test whether or not a population is in Hardy-Weinberg equilibrium. And if it isn't, that means that at least one of these criteria is being violated. Either the population is too small, or there is selection going on. Haha, that's interesting. Or because, you know, that's what we study. You know, would be an interesting find. Or there are high levels of mutation, although realistic mutation levels don't do very much to move a population out of Hardy-Weinberg, but maybe the population is like directly downwind from Arkansas 1 or something. Or mating isn't random, so maybe we've got inbreeding or sexual selection, or there's migration in and out, or maybe there's several of those things going on at the same time. So if a population is not, we can test whether a population is in Hardy-Weinberg, and if it isn't, that means something interesting is happening that's causing that population to change. And we can try to figure out what that something might be. It actually gives us a pretty good null hypothesis. What we're going to do in some of these practice problems, and in a real world lab that we're going to do this Tuesday, what we're going to do is estimate genotype frequencies if a population's in Hardy-Weinberg, and use good old-fashioned chi-square to compare the data from the real population with the data that we would predict if the population is in Hardy-Weinberg. And, well, then we can test whether the population is likely to be in Hardy-Weinberg or not. And if it's not, then one of those 5 is being violated and something interesting is happening to the population.
So it's pretty hard for population to be.
Well, there's no population that's in perfect equilibrium because, I mean, if it was, it would have to be infinitely large. You're always going to get slight little deviations off of Hardy-Weinberg equilibrium. It's just one of those things that happens. But is the deviation big enough to be statistically significant is usually the question that we ask. So yeah, there's no population that's perfectly there, but we have populations that are close enough. And then we have populations that are definitely nowhere close. One little postscript, Hardy was actually kind of proud that the math that he worked on had no obvious real-world applications. In his autobiography called A Mathematician's Apology, he wrote, "I have never done anything useful. No discovery of mine has made or is likely to make, directly or indirectly, for good or ill, the least difference to the amenity of the world." In context, by the way, Hardy was writing this at the beginning of World War II, when a lot of mathematicians and physicists and other scientists were doing work related to the war effort, were using their math and their physics knowledge to develop powerful weapons and defensive things like radar and, ultimately atomic weapons. Hardy was kind of comforted by the fact that his work was so abstract that nobody was ever likely to use it to develop a weapon. And nobody has, although a lot of number theory revolves around prime numbers, and some of Hardy's work on primes is actually used in internet security and internet coding. Ways to encode data and send it over the internet securely involve using extremely large prime numbers. The most common type of encryption called PGP makes use of the properties of primes, and I'm not even going to try to explain why. But the fact that you can buy things off Amazon and more than half the time nobody steals your credit card data is made possible by application of number theory. So some of Hardy's work was useful, but his best known achievement was this little piece of elementary algebra that he just knocked out over breakfast. It's certainly not what he spent most of his time on. It's certainly not what he would want to be known for. And yet it has made a great deal of difference in the amenity of the world. And starting on Monday, we're going to find out what kind of difference it actually has made. And we're going to start working on actual problems. By Monday, actually before, there will be a sheet of practice problems up on Blackboard. Starting Monday, start bringing your calculators. You're going to need these for the exam. I'm not going to let you use your phones because, sorry, I'm not going to let you have the ability to surf the internet while you are taking an exam. But remember, you're going to need a calculator, and it doesn't need to do anything fancier than taking square roots. So those are what, like $10 or something like that. You may already have one. Shoot, these days you can probably get them as prizes in your Cracker Jacks or something. So have one of those handy. And we're actually going to get away from using slides for the next few lectures. And I'm going to start working problems on the board. So be prepared for that. I will go ahead now and stop the recording.